All right, so I'm Dr. Wassif, and I'm going to be taking Bob's approach, and I'm actually not going to stand up behind a podium because I don't do that very well anyway, and I tend to wander around a lot. So, as Gretchen mentioned, I'm at the National Institutes of Health. Dr. Porter's senior staff scientist in the laboratory, and as such, I'm going to be talking about the preclinical models, specifically zebrafish and mice for the study of Smith-Lemli-Opitz syndrome. So why in the world zebrafish? Who cares about zebrafish? Why were we thinking even about jumping into zebrafish? Zebrafish are a unique model system in that they are completely clear as they develop, so it allows you to actually image the brain as it is developing over time. And from a single mating, you can typically get 200 to 300 embryos at a time, which then obviously gives you genetic capabilities as well as power in order to do major studies. Whereas mice take a ton of space and a lot of money to house, the zebrafish don't. So our goal was to generate a viable model of Smith-Lemli-Opitz syndrome in the zebrafish. We were hoping to be able to study the developmental and functional neurological defects of the accumulation of 70-hydrocholesterol in the brain of these fish. And then ultimately, it would give us a powerful means to be able to screen drugs, because when you've got 200 or 300 fish, obviously it's pretty easy to start testing lots of different compounds at lots of different concentrations. And I'm not going to bore you with all of the details of how we went about making the fish, because who cares at the end of the day. I'll let you know that yes, in fact, we did generate the SLOS fish, and that in many ways it mimics the human syndrome in that you see decreased levels of cholesterol and elevated levels of 70-hydrocholesterol. You see that in all tissues, but we tend to focus mostly on the brain. After all, that's where most of our interest lies, in that how can we address the issues of intellectual disability, as well as the behavioral phenotypes, as well as in the liver, because the liver is where most of the cholesterol in the body gets made. And as you can see, we do have a significant decrease in the amount of cholesterol in both the brain and liver, and undetectable amounts of 70-hydrocholesterol in the wild type as well as the heteros, but significant amounts of 70-hydrocholesterol in these mutant fish. We made 2 separate lines of these fish so that we know, in fact, that we didn't accidentally hit someplace else in the genome, and that everything looks the exact same. These fish are much smaller than control or normal or heterozygous fish. You can see here that they are, in fact, significantly smaller. You would almost refer to this as failure to thrive. And I know that the clinical people don't like that term anymore, but these fish just don't grow quite as large as the regular zebrafish. They also die earlier than their control litter mates. And so just a quick recap, because we're still in the early characterization of these. The fish recapitulates the model of Smith-Lemli-Opitz in that biochemically they have increased levels of 70-hydrocholesterol. They have significant growth retardation, and they die prematurely. Our goals are pretty straightforward for this fish at the moment. We're going to continue on with the characterization to identify exactly what's going wrong with these fish, and to then hopefully be able to use this model system to look at both mechanism as well as therapeutic interventions for SLO. So I'm going to jump a little bit now to where I've been focused for many, many years, which is the mouse model of Smith-Lemli-Opitz syndrome. And I'm going to talk specifically about modifier genes and what does that exactly mean. So what is a modifier gene? And I called my 14-year-old son last night because I needed to check in on him anyway, because I was working till 2 o'clock in the morning trying to get all this put together. And I tried to explain to him what a modifier gene was. And so I gave him the clinical definition of what a modifier gene is. And I told him, it's a genetic modifier that whenever it's expressed, whether it's a lipid, whether it's another gene, it's going to have an effect on other genes within the genome. He went, "What are you talking about, Dad?" Well, I went a little deeper and I said, okay, modifier genes can affect the transcription of other genes. They also can affect other things up and down throughout the genome. And he still looked at me like I had 3 heads. And so I broke it down and made it simple. So hopefully I've made it simple for everybody in the audience. So I started with a vanilla milkshake, simple vanilla milkshake. Made it on the counter, no big deal. I added a modifier gene. In this case, all I did was add red food coloring. Well, that red food coloring could be any other gene within the human genome. Well, I'm still left with just a vanilla milkshake when I'm done, but it looks different. Obviously, we went from having a nice white milkshake to a red milkshake. That's essentially what a modifier gene does in many aspects. It changes the phenotype of the presentation of that disease. And we have an interesting system in the lab currently. We have the two most common mutations represented in the human population present in our mice. We created a nonsense mutation, or a full null, in that these mice have no activity whatsoever. And we have a missense mutation in the T93M, which is the most common missense mutation we found in the human population. When we place that exact same set of mutations on the black 6 background, and black 6 is an isogenic, and just means every single one of those mice are the exact same. We put that on a black 6 background. Those mice are 100% lethal. Every single one of them die at birth. We place those two exact same mutations on FEB, and those mice 100% live. They have a normal lifespan, they run around, perfectly healthy, happy mice. Well, why in the world is on one background those mice dying, and why is one set of mice perfectly fine? So in order to get at why one survives and one dies, we employed basically this strategy where we're looking for modifier genes. And what we simply do is cross those two isogenic strains, the black mice and the white mice. Juki, you're welcome to hang out with me. Not a problem, buddy. In fact, you can give the talk. So we cross those black 6 mice and those FEB mice, and we look at the ratio at which mice survive versus what ratio they die. And when we cross these mice, typically, when we add 50% of black 6, 50% FEB, those mice 100% survive. They're running around, life is good. We then do a second cross, and we add additional black 6, so we back cross to another black 6 mice. So we're just simply increasing the amount of genetic material that came from black 6. And those mice start to die. We're now at 75% black, and we're starting to see a little bit of death. We back cross one more time, and just to go ahead and get to the data so that you understand what I'm showing you. When we actually get to the point that 75% of the genetic background is black 6, we're starting to see increased death in those mice. So there's a hint that there's a gene present in black 6, which we kind of already knew, that is going to affect whether these mice live or die. And so there are ways that you can look at SNP arrays and look throughout the entire genome to focus in on what gene or genes are truly involved with that survival versus death. And we're still in the process of putting all that data together, and I'm hoping in the next year or two we'll be able to present that data to you to be able to say, hey, these are where this is. Hopefully at this point we know sort of which chromosomes to look at, but right now it's still all really, really preliminary, so I don't want to get into the details in full amounts. But it looks like there's probably a single gene. We're excited about that. We're continuing to do the map. We're now doing what's referred to as fine mapping to try and get in closer and closer to the loci, but still have a long ways to go. We've been collecting the pups that survive as well as the pups that die with the idea that they both provide you a different set of information. And hopefully this provides some insight into both mechanism as well as potential other therapeutic interventions. If we have another gene that we know can affect survival, is that a gene or a pathway that would be worth upregulating or targeting with some kind of therapeutic compound for the treatment of SLO? And the last project that I'm going to talk about before I hand off to Simona is a project, again, looking at modifiers, but this time we're looking at modifiers of sterols. And the typical pathway for cholesterol or excess cholesterol to be removed from the body is through bile acids. But you also have a whole set of oxidative cholesterol products. 7-dehydrocholesterol can enter into those pathways as well, either through enzymatic methods or through non-enzymatic methods. And you get the accumulation of a whole series of both normal as well as abnormal oxysterols in these SLO children. And we focused in on one specific oxysterol, mostly just because of the availability of a transgenic mouse. And what we looked at was CYP27. CYP27 normally converts cholesterol to 27-hydroxycholesterol. Well, it's a very promiscuous enzyme in that it's perfectly happy to work on 7-dehydrocholesterol. And it converts 7-dehydrocholesterol to 27-hydroxy-7-dehydrocholesterol. And there's been work by Phil Beachy's group as well as Ned Porter's group looking at the effects of these oxysterols. And as you're going to hear a lot tomorrow in Alan's talk, there's growing evidence that the effects of oxysterols as well as oxidative stress in SLO is probably significantly bad, for lack of a better term. And so when we generated these pups, we found out very quickly that, in fact, the changes in the biochemistry resulted in a significant change in the phenotype of these mice. Both mice, the full null as well as these mice that overexpress CYP27, die at day of life 1. But as I hope you can tell from these pictures, these guys that now overexpress CYP27 are significantly smaller and significantly more dysmorphic, as I'll show you in some other slides, but hopefully shows up relatively well here. They have micronathia, or the small jaw. It's slightly hidden by the pup's paws, but they have umbilical hernia, they have a distended abdomen, they have all sorts of polydactyly, postdactyly, polydactyly, they have 2-3 fusion, they have all sorts of limb defects, some of which are present in, obviously, the SLOS children. So what happened with the biochemistry side of things? When we looked at 27-hydroxycholesterol, there was no correlation. When we looked at the levels of 27 whoa, hit the wrong button. My bad. When we looked at the levels of 27-hydroxy-7-dehydrocholesterol, there was a significant correlation in the amount of cholesterol present. So as these oxysterols increased, the amount of cholesterol was decreasing. So that fit very well with a lot of the clinical data that we had, in that as we looked at the amount of 7-dehydro and 8-dehydro combined, i.e. the dehydrocholesterol, we noticed that there's a significant trend towards higher levels of dehydro resulted in lower levels of total cholesterol as well as total sterols. So this animal model may start to explain some of that, in that these increased oxysterols are potentially shutting down total cholesterol synthesis. Just going back to the mouse a little bit more, it's significantly more dysmorphic. It actually more closely mimics the patients than the original mouse model, in that about 77% of these mice present with cleft palate. They all have micronathia. They all had umbilical hernia. We see limb defects in about 50% of all the limbs that we studied, and they all have this kinked, curly little piggy tail. Other manifestations we noted was ventricular dilatation. We've noticed agenesis of the anterior commissure, reduced cortical plates. There's all sorts of lingual and dental problems, as well as cardiac malformations. So this model system is allowing us to both study the dysmorphic features, some of which are very much present in SLO, such as cleft palate. So it's allowing us to look at some of the mechanisms that are present that causes those defects to occur, as well as a whole host of other defects that we haven't been able to study previously in the original mouse models.